Ladies and gentlemen, dear colleagues, gene therapy drugs, a breakthrough in the treatment of genetic diseases. The global healthcare system has to deal with the difficulty of diagnosis and lack of effective treatment of many genetic diseases. Without appropriate treatment, they lead to death or disability of a person. For such patients, medical care is often limited to symptomatic and palliative treatment. Since the 2000s, gene therapy drugs have been a promising direction for the treatment of these diseases. In this talk, we'd like to present the review on the use of gene therapy drugs for some monogenic human diseases. We'll start with some information on the types of gene therapy drugs and then focus on one medical condition for the treatment of which a gene therapy drug can be used. Well, the gene therapy drugs allow for precise effects on a specific gene due to the emergence of new genome correction technologies. Most modern gene therapy drugs perform genome correction in vivo. However, the use of vector constructs and genome editing systems makes it possible to eliminate a gene with a mutation or insert a copy of the correct gene to the genome of cells previously isolated from the donor's body. This is referred to as ex vivo gene therapy. Such drugs are used in the treatment of genetic diseases for which there are no available treatment methods or drugs aimed at eliminating the cause of the disease. They are also used for the treatment of oncological diseases and life-threatening conditions. The development and conduct of preclinical and clinical trials tend to be a more complex, time-consuming, long-term, and expensive process compared to traditional medicines. Currently, a drug based on genetically modified human cells have been approved for the treatment of severe combined immunodeficiency. It is Strimulase. This ex vivo gene therapy drug was used to treat severe combined immunodeficiency caused by deficiency of the enzyme adenosine deaminase, ADA. Mutations in the gene encoding the ADA enzyme lead to the development of the disease, which manifests itself in a decrease in immunity and the appearance of complications associated with metabolic disorders. Before the advent of gene therapy drugs, the only way to treat this pathology was allogeneic bone marrow transplantation from relatives of the recipient with an identical major histocompatibility complex, or replacement therapy with bovine adenosine deaminase. This ex vivo gene therapy drug for the treatment of combined immunodeficiency is based on the use of the ADA enzyme gene transfer system, using a retroviral vector into peripheral blood lymphocytes. At the same time, despite noticeable improvements in the condition of patients, the level of the ADA enzyme in their blood was insufficient. Therefore, the transfer of the enzyme gene began to be carried out in pluripotent hematopoietic stem cells. That made it possible to significantly increase the number of cells with the corrected genome capable of proliferation and transfer of the genetic construct to daughter cells. The subsequent improvement of the gene therapy procedure led to long-term remission of the disease and the termination of any treatment in patients. Therefore, in 2016, the European Medicines Agency approved the commercial use of Strimulase to patients without a compatible donor. The drug cells are transplanted into the patient's bone marrow, where they express the active enzyme ADA. After successful cell engraftment in patients, a lifelong therapeutic effect is possible. As a result of a 3-year open clinical trial, a total of 18 patients of different races under the age of 6 years underwent this treatment. The therapy resulted in 100% survival of patients after 7 years, a decrease in the severity of concomitant infection diseases. 7 people didn't need immunoglobulin therapy for 3 years. To sum it up, the genetic modification of cells isolated from the body may consist in correcting a defective gene or knocking it out, as well as introducing a correct copy of the gene into the cell. It is the use of gene therapy drugs that will achieve a long-term effect and stable remission of genetic diseases. Further development and improvement of such drugs will make it possible to abandoning substitution therapy or palliative treatment, as well as significantly prolong the duration and quality of life of patients. Thank you for your attention.